Okay, thank you very much for the kind invitation and the opportunity to present here our personalized Swiss sepsis study in behalf of our SPHN and PHRT driver team. I'm responsible for the SPHN part, and Carsten Borgward from the ETH Zurich is responsible for the PHRT part. Now, but we are really a huge team, and I will introduce you later on the different players. And I would really like to thank all the collaborating partners at this point. So far, there were fantastic discussions, and I really learned a lot already. Now, let me start with what sepsis is. Sepsis is a syndrome and not a single disease, and this definition is somewhat challenging in the clinics to recognize patients who have a sepsis. Sepsis is, however, a very frequent clinical problem. There are thousands of cases every year in Switzerland, and it normally starts with a bacterial infection progressing then to sepsis. And this is associated with a substantial morbidity and mortality. In certain groups, it can really reach up to 50–70%. Associated with sepsis is a reduction of the quality of life if you survive sepsis. And these are certainly the most important impacts for the individuals: high morbidity, the mortality, and the reduction in quality of life. But as a society also, it is very important to understand that these critical ill patients, they need a lot of healthcare costs, especially due to their need of intensive care medicine and work life basically being lost. Now, the definitions of sepsis evolved over time. So about 20 years ago, sepsis 2 definitions were generated, and what is a hallmark of sepsis is a proven or suspected infection, and the SIRS criteria. And SIRS stands for Systemic Inflammatory Response Syndrome. So this is an inflammatory response from the host towards the pathogen. But it can also, all sorts of diseases can generate an inflammation. So some of these factors, for example, body temperature or a high heart rate, I will actually get if I walk here to the first floor or second floor. I will fulfill certain criteria of SIRS. So it is a very sensitive definition, but not necessarily a very specific one. So this was the reason why the sepsis definition was adapted a couple of years ago to the sepsis 3 definition. And here you can see again, the infection is very crucial, but now you have different elements which are monitored, the so-called SOFA score. And SOFA has nothing to do with the couch. It's actually a sepsis-related organ failure assessment. And this monitors different organs in a patient. So for example, here you have respiratory measurements, you have mental status, which is assessed, then you have blood pressure, laboratory values, like for example, hepatic function, kidney function, and hematological functions. And this helps us to better identify patients with a sepsis. So this is a very specific definition now, but also with this kind of definition, you might miss some patients who have a sepsis. So it's really a problem with sensitivities and specificities. As I mentioned here, inflammation, and this SOFA score is basically assessing the risk for mortality. Okay, so the course of sepsis is highly heterogeneous, and it depends on 3 important factors. First of all, the pathogen. So there are roughly about 10,000 known bacterial species. Not all of them are pathogens. But you can imagine that a pathogen brings a lot of diverse factors into this interaction. Virulence factors like toxins, a pathogen can also become resistant to certain treatments. So it's really adapting to its treatments and the host. Then you have the patient, and we are very diverse here in the audience. You have different age groups, different genders, then genetic backgrounds, and also the way how we respond from the immunological system is also very diverse. And the third factor, which is crucial, is the timing. So if somebody presents early on in sepsis or very late with a progressed sepsis, that makes a difference in the outcome. And this was very nicely summarized by a study from Kumar and colleagues about 10 years ago. You can see here on the x-axis the time from hypertension onset. So this is basically shock, the duration of shock. And you can see here the longer it takes to establish antibiotic treatments, this is the bright gray bars, the lower your chances are to survive. So early recognition is crucial, and especially the identification of the bacterial pathogen makes a difference there for a successful treatment outcome. And this is a study we've done in Basel together with the clinics for infectious diseases. I would just like to highlight how important it is to know what pathogen causes a sepsis. You can see here a time scale, time since blood cultures were drawn in hours. Here the blood culture is taken, and we had two different types of identification methods to identify the causing pathogen. Conventional identification, so you wait basically until the blood culture gets positive, you do a gram staining, then you take blood from the positive blood culture, you subculture it, and then the next day you make an identification. That takes about 55 hours, and then after a while you have antibiotic susceptibilities available. Now, in this new technological arm, we use the method, a mass spectrometry method, to identify directly in the positive blood culture the bacteria. So the time to gram staining is the same, but the time to identification of the bacterial species was about 24 hours faster. The susceptibility information still takes time because the mass spectrometry does not provide you with information on the susceptibility directly. Now, on the right graph, you can see again the hours since the blood cultures are drawn and the cumulative optimal treatment. And this is here an example of personalized bacteriology. You can see here the bright gray arms here. This is multitoph and conventional identification. For Escherichia coli and Klebsiella, it didn't matter if you knew early on what it was, because there you need a specific susceptibility information. But for other bacteria which carry intrinsic resistance information, there's a huge difference if you know early on what it is. So the consequence of this identification was that we had a reduction in the time of identification, we had an optimal treatment at an earlier time point, and the 30-day mortality in the patient cohort we examined, and these were about 200 patients per arm, there was a substantial reduction in the mortality. So what I've shown you now is basically an introduction into sepsis. So what is important? There are three factors: the host, the pathogen, and the timing. And in the hospital, lots and lots, we've heard it this whole day, lots of data is collected. So we will collect data, and we envision basically that by collecting healthcare-related data like clinical data, laboratory data, and omics data, we will understand sepsis better when we use novel interpretation algorithms. I think the complexity of sepsis patient is really unimaginable due to these three factors, and I think we really need a different way to approach this huge amount of data. We think that we will understand this complexity on a better level, that we will also develop and found new types of biomarkers, maybe also which physiological compartments and what time points should we sample to also save diagnostic costs and thereby improve the outcome of the patient. So early recognition to then reduce sepsis-related mortality. And I think it is very important to make the data interoperable, as we have heard here now multiple times. it's important that we can share data between centers to reach sufficient patient numbers. And I think basically this will lead to a framework where personalized diagnostic and therapeutic research for infectious diseases and intensive care medicine can be performed. Now, as I mentioned, many people are involved in this. So from every university and university hospital, from Basel, Bern, Lausanne, Geneva, and Zurich. On every center, we have an infectious disease specialist. We have an intensive care medicine expert. We have the clinical microbiologist on board. So this provides basically the baseline clinical expertise on this complex disease. But then we also have researchers from the biocenter, statisticians, machine learning experts, and omics specialists from the ETH, really completing this kind of study team. So what is the framework we would like to build up in this study? So every university hospital will build a data warehouse, independent of this driver project. What the goal of our driver project of the SPHN part is to connect the intensive care units with the data warehouses, to collect data here, bring this here. Also laboratory tests and clinical tests focusing on sepsis. And then to share this data via the Data Coordination Center with the PHRT part where basically machine learning algorithms will be used to understand that. But that should not be a one-way direction. The results then should be put back into the data warehouse and help us to really improve the quality of the data, first of all, which goes down here, but also to learn and use these biomarkers on the go. Then we have also an omics technology part, which comes from inside the ETH, but also lots of clinical partners, laboratories, which will provide here respective technologies. So what are the two project parts? What are the specific goals? For the SPHN, we try to set up the infrastructure to connect the Swiss university hospitals, in particular the intensive care units, then to define ethical and governance standards. I think this is very important to do, and then basically make the data sets interoperable and comparable. And how we organize that is in different work packages. So ethics takes a huge part, and the governance structure, that's actually over the 3 years. This is a 2-year time frame where I think we will need lots of discussion and adaptations. Then the requirement analysis, which will shortly start, or we are already starting actually, to see what kind of data do we need to collect from the intensive care units. And then the IT specifications, the target architecture, and the interface towards the data warehouse will be designed. And then we can, once the data warehouses are filling up with data, we can compare data in between the centers and build a connection. Now, for the PHRT, I think collecting data is a nice thing, let's say, but without a research question, it's kind of useless. And I think it's also very, very important that we build and test the infrastructure with a concrete research question. And what we aim to do is early recognition of sepsis, prediction of sepsis-related mortality. And with this data, we try to discover novel type of biomarkers, and there are basically 3 types. Digital biomarkers, and this involves time series, for example, temperature profiles, blood pressure, and routine laboratory parameters, but not at a single time point, but really in a multi-dimensional fashion over time. Then the molecular biomarkers, this will include, and I just put here a few examples, metabolic profiles, pathogen evolution we have, then certainly also immune phenotyping. The selection of the patient's time points and compartments I mentioned before, I think it's very crucial to learn which compartment and what time points we need to answer these questions. Maybe it doesn't make sense to have a certain time point, and then we should really omit that for the future. And then also giving back the feedback to the SPHN part. And this is how the work packages are organized, digital biomarkers, the molecular biomarkers. We have here various omics specialists, metabolomics from Professor Zamboni, then immunophenotyping and human genetics from Professor Calandra at the SCHUV, shotgun metagenomics from Professor Groib and Professor Schrenzel, and myself, who will look into bacterial whole genome sequencing. Then we try to generate hybrid biomarkers, so a combination basically of both worlds, digital and molecular. And then the last point will be the reproducibility. Are there certain differences between the centers? Now, what do we envision with this joint SPHN and PHRT proposal? It's clearly, this is why we do it. We do it for patients. There should be an impact on the individual patient, which promises to be drastic because the mortality rates are really dramatically high, and we hope that we can further reduce that. Then highly innovative possibilities for research partners, but also maybe pharma and diagnostic companies, which can take up certain biomarkers, and we can really share the knowledge we generate. Then frankly, I hope that we will be the best global sepsis data collection on the globe. Currently, there are a few such studies going on, especially in New Zealand and Australia, but I think with the Swiss quality brand, we can be there very competitive. And then what was also mentioned this morning is the sustainability. This is absolutely key that we find a way to go beyond 2020, because once the infrastructure is built up, we will certainly not stop, but we should then use this infrastructure and do further research. And I hope that this will put Switzerland then at the forefront of sepsis research. Now, as I mentioned, I'm responsible for the SPHN part, Carsten Borgward for the PHRT, but at the end, it's a whole team of people, and here I have listed all the investigators, and I would really like to thank some of you are in the audience. It's, I think, a really challenging journey ahead of us, and we will need a lot of discussions, and please, if you have from a particular center some questions, you can directly address these people. So thank you very much for your attention.